I'm going to talk about the secretome of apoptotic peripheral blood cells and its role in the microvasculature in acute myocardial infarction. The talk will be divided into two sections. The first part will be about the role of platelet aggregation, and the second part will be about the role of secretome in vasodilation. I personally have no financial disclosure. However, the Medical University of Vienna has claimed financial interest in regard to the data I'm just presenting, and the senior author of this project is a shareholder of a company which also has claimed financial interest. As you know, during acute myocardial infarction, we are sometimes confronted with a so-called no reflow phenomenon, which is associated with the microvascular obstruction, which means that despite the fact that the infarcted vessel is quickly reopened, we still have a disordered microvascular perfusion and an inadequate perfusion at the level of the small vessels. This is more or less a multidimensional process with two key players. The first one are platelets, and the second one is the microvascular vessel itself, which can be spastic during this microvascular obstruction. On the right side, you see a model of a porcine heart, and all these small reddish fibers correspond to small capillaries, and which show you that these small vessels are essential in providing the heart muscle with nutrition and with oxygen. So what is APOSEC and what lessons have we learned so far from secretome analysis? There is quite good evidence so far that acute myocardial infarction can be very effectively treated with secretome of stem cells. This is the so-called conditioned medium hypothesis, which was first formulated in 2005 and very prominently published. But only 3 years later, it was found out that the secretome of stem cells is in fact quite similar to the secretome of peripheral blood cells and contains comparable levels of cytokines and chemokines. So we formulated an hypothesis that the secretome of irradiated peripheral mononuclear cells could be effective in quite the same way as the secretome of stem cells in regard to ischemic diseases. APOSEC, which is an acronym for the secretome of irradiated peripheral mononuclear cells, is nowadays produced in a standardized manner, starting with a venous blood collection. Then PBMCs are separated through fecal density centrifugation step, and the PBMCs are then, after irradiation, incubated for 24 hours. Then the supernatant is separated from the cellular compound. The supernatant is stylized and lyophilized and can be used in different experiments thereafter. We have previously shown that APOSEC is in fact very effective in attenuating the myocardial injury following acute myocardial infarction. So the aim of this study is to look at the role of APOSEC in regard to microvascular obstruction. We applied a closed chest acute myocardial infarction model of the pig, where a balloon was inflated in the LAD for 90 minutes. Half of our pigs received APOSEC intravenously, half of our pigs received the control medium. After 90 minutes, the balloon was deflated again, and the pig was observed for 3 months, and there were different time points for measuring all kind of factors. This in vivo model was supplemented with a bundle of in vitro experiments looking at the role of platelets and looking at the role of microvasculature. Namely, we performed aggregometry experiments, flow cytometric experiments, we applied ELISA technique and Western blot experiments, and for the second part, we performed coronary ring assays, also ELISA, Western blots, and immunohistochemistry. So this is the first result slide, showing you that in fact APOSEC is effective in reducing the area of microvascular obstruction. This can be very easily visualized 3 days after acute myocardial infarction when looking at hypoenhanced areas in the so-called late enhanced MRI images. And if you look on the right side, we could show that APOSEC in fact significantly reduced the area of microvascular obstruction in our model. And this was associated with findings in the heart cath examination. Here you see 2 examples. On the right side, an APOSEC treated pig, where the contrast agent very quickly flushes towards the periphery, whereas in control pigs there is not a quick flush as observed in the APOSEC pig. And this can be, of course, evaluated and measured objectively in the so-called corrected TIMI frame count, which is significantly better in APOSEC treated pigs than in control pigs. Additionally, the myocardial blush grading, this is another method to evaluate microvascular obstruction in the heart cath lab, was also significantly better in APOSEC treated pigs than in control pigs. And these findings were also associated with differences in regard to ECG monitoring. Only 1 out of 7 pigs had an ST resolution after the procedure in regard to the control pigs, but 4 out of 6 pigs had a complete ST resolution in the APOSEC group. When looking at Holter ECG analysis, we found that episodes of ventricular extrasystoles, couplets, triplets, or ventricular tachycardias were observed less or in few number in the APOSEC group when compared to the control group. So we took 1 step back and looked at in vitro experiments, and these are data performed with highly purified platelets, and those purified platelets were activated with either TRAP6 or ATP, and the clotting was measured, and by adding APOSEC, the clotting of platelets could be significantly reduced in a maximal and in a half maximal dose of TRAP6 and ATP. And this was associated with a reduced expression of activation molecules on the surface of platelets, namely CD63, CD62P, and CD40 ligand. But not only the surface proteins of activation were decreased, also the secreted products, platelet activation were reduced. Here you can see data from soluble CD40 ligand and the soluble P selectin, together with thrombospondin 1, which is a novel marker of platelet activation. Also thrombospondin 1 was significantly reduced when adding APOSEC to activated platelets. So what is the mode of action behind this? We found out that the VASP phosphorylation was significantly enhanced by APOSEC. VASP is a pathway which is responsible for the change of conformation in resting platelets when they are driven towards activation. And here you can see that by adding APOSEC, the amount of VASP phosphorylated platelets significantly increases, and this effect can even be augmented by adding the natural agonist of this pathway, PGE1. So, getting back to our in vivo experiments and porcine AMI model, we could observe that in the serum of pigs, the soluble P selectin, the thrombospondin 1, PF4, and soluble CD40 ligand was significantly reduced in APOSEC treated pigs when compared to control peaks. But getting back to what I've previously said, that microvascular obstruction is a multidimensional process, we were also interested in another factor, whether APOSEC could reduce microvascular spasm. And these are again data from serum samples of the pig experiments. Nitric oxide was significantly high in APOSEC-treated pigs, PGE2, and VIP was significantly higher when looking at control pigs. So this means there might be some APOSEC-mediated vasodilation in our model. And when looking again at in vitro experiments, we found out that highly purified endothelial cells, the Hubeck culture, exhibited an increment of INOS expression and an increased phospho-INOS expression 60 minutes and 24 hours after treating those cells with APOSEC. And this was associated with findings from isolated coronary rings, which were incubated again with APOSEC, and you see that on the right side of this slide, the phospho-INOS expression was significantly higher in the treated rings when compared to control rings. All those effects were, of course, NOS dependent. However, we also observed direct NOS-independent effect in so-called myographic experiments. You see that when adding APOSEC to coronary rings, the maximal wall tension was within seconds reduced by about 20%. And why this is NOS independent, you see below. By adding an NO blocker, this effect was still observed. And this effect was, of course, dose dependent, starting from 1 to the 3 to 1 to the 7. So, all in all, what functional effect has this reduction of microvascular obstruction in our porcine experiments? We performed MRI analysis 30 days after AMI induction, and you see that APOSEC-treated pigs had a significantly increased ejection fraction, whereas the area of infarct was significantly reduced. So to conclude, we could show that APOSEC reduced the areas of microvascular obstruction during acute myocardial infarction, and this was due to an inhibition of platelet activation and platelet aggregation. This was due to an induced vasodilation, and this finally led to myocardial salvage. Thank you very much for your attention.